Hello and welcome to Smith and Sheridan on Biotech, a podcast on the science and business of biotechnology. It's presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, everyone. Hello, Cormac.
Hey, Andy, how are you doing?
Fine, thanks.
You wanted to talk about CAR Ts this week, or to give them their full name, chimeric antigen receptor T-cell therapies.
Yeah, and I'm glad you've gone for that one. Also, I'm hoping that later in the conversation, you're going to attempt their full generic names as well, but I'll probably stick to the brand names. But CAR Ts, I don't know if you felt this when you first read about them, but it's almost the stuff of science fiction, isn't it? That you could take a cell or some cells from a cancer patient, reprogram them, activate them, amplify them up, and then get them back into the hospital in time to infuse into the patient to, in some patients, result in a cure of their cancer. I mean, it's fantastic, isn't it?
I remember, and I haven't checked back, I have to confess, but 2012 was the first time I heard about these therapies that were still barely in patients, if in patients at all at that stage. I guess they were just about doing some early trials. And I was writing about something else, and a very smart guy brought them to my attention. And I'm glad you used the word about reprogramming, because I remember covering the 2017 FDA Advisory Committee meeting for the product that became Kymriah. I'm not going to attempt the generic name either. And I remember writing a piece about it, and I had that same idea, the idea that this was before the approval, but it was pretty obvious it was going to get approved because of its spectacular efficacy. But it showed the fact that T cells were programmable. And that to me was something that, and programmable in ways that actually had very meaningful clinical outcomes. It wasn't something that it might work in 5% of cells in a lab and grown in some sort of a flask. This was actually cells that you took from a patient. You use some sort of a viral vector to transduce those cells to transfer in a construct, a DNA construct that encoded a kind of a Heath Robinson contraption that altered the specificity, the binding specificity or the recognition capacity of the T cells. And obviously, the T cells did retain their inherent binding affinities as well as this new affinity, but those new affinities were strong. And just for people who are not familiar with what they consist of, they contain an antibody fragment that recognizes a protein expressed by a cell type of interest that you're trying to kill or control in some way or in some way modify. And then it's fused to typically two, and this is expressed on the outside of your programmed cell, and then that's connected up with linkers to two signaling domains that are within interior to the cell. And one is to ensure that that T cell becomes activated and expands when the bit on the outside binds to what it's supposed to bind to. And then the other bit is to ensure that they expand and persist. So typically it's a CD3 zeta domain for the T cell activation piece, and something like a 41BB activation domain or persistence and enhancement domain, or maybe CD28, these co-stimulatory domains.
And we'll probably get round to the benefits and some of the Achilles heels of CAR T therapies, but yeah. I wanted to add that point to add color to your description, Cormac, in that if you're a microscopic organism that's foreign to the body, or even if you're a foreign antigen, and you don't want to encounter an activated T cell because they are pretty aggressive things. And it's amazing that you can then take the theory, the science, and reprogram him to do what you want in the body externally.
And what's also interesting to me is that, I mean, the first rudimentary CAR Ts were described as far back as the 80s. Researchers in Japan and in Israel, I think, are credited. There's a very good Wikipedia article about it if people want to check it out. But these concepts were being explored quite a long time ago. And what's also interesting to me is that the first efforts to actually test them in patients didn't involve cancer patients at all. They involved people with HIV-1 infection.
Yeah, that's a great step back down memory lane.
Yeah.
I can remember a JP Morgan conference as an investor. It's decades ago where I saw the company, or one of the companies that was doing that, activated T cells reprogrammed against HIV. I thought, this is a wonderful idea. This is great. And I never heard of the company again.
Cell Genesis, I think, was the kind of leader in that area.
Mm.
And Kirsten Hege was their person, the clinician who kind of led it. But interesting enough, an early collaborator there was Carl June, now at UPenn, but he was then at one of the NIH institutes, and he was a collaborator. And I think the problem with the early ones was that they didn't have this co-stimulatory domains, and they were themselves susceptible to HIV infection, being T cells, even though they were typically CD8 cells, but they probably would have had CD4 cells mixed up there too. But interesting enough, one of the early researchers involved in that, Boro Dropulic, who was involved in different types of work around HIV, is now involved in a not-for-profit organization called Caring Cross, and they're continuing CAR T story in HIV with a dual targeting construct, targeting two different epitopes, fragments of the HIV glycoprotein 120 protein. I spoke with them recently, and they just want to really zap the infection very hard. And if there's any kind of latent virus re-emerging from those infected cell reservoirs, that they'll hit it hard. And I think in their clinical trial that the participants will be taking a holiday from their antiretroviral drug therapy. You really get a chance to see whether or not there's going to be viral rebound and whether or not this therapy is going to be adequate to the task. And they said themselves it may need to be combined with other things. But we then come to Kymriah, and there was a whole sort of a plethora of other products that followed, and they all target CD19, funny enough. And I remember speaking again between the AdCom and the approval of Kymriah, speaking with one of the early pioneers of CAR-T development, Rainier Brentians, who was a Juno Therapeutics co-founder. He was then at Memorial Sloan Kettering Cancer Center in New York. He's now at the Roswell Park Comprehensive Cancer Center. And he said to me, and I quoted him in the article, and I'm quoting him now, he said there was a lot of excitement around what was happening, obviously, with the data. And I felt he was slightly raining on everybody's parade, but I think his words were really prophetic. He said, have we developed a new treatment for B-cell malignancies, or is this a new paradigm for cancer in general? And it's no coincidence that everybody's going after CD19. So we have 4 CD19 targeting CAR-T therapies approved now, as well as Kymriah from Novartis. We have Yescarta from Kite Pharma, which Gilead bought for 11.9 billion, purely on the basis of that, more or less. Kite Pharma has a second CD19 product called Tecartus, which is just for a slightly different set of lymphomas and leukemias than what Yescarta addresses. Bristol Myers Squibb, having picked up Juno via its acquisition of Celgene, has Breyanzi, which is also CD19 targeting. It's for large B-cell lymphoma. And then we've got 2 more recent approvals for multiple myeloma. They both target B-cell maturation antigen, Abecma, which BMS developed with Bluebird Bio, and Carvykti, which Johnson & Johnson or Janssen developed with Legend Biotech. And that's it. So in this revolutionary therapeutic modality that gained its first approval 6 years ago, we have a grand total of 6 approved products. And there was a huge rush and huge efforts to extend the concept across multiple areas of cancer and non-cancerous indications, which we'll come to in a minute. But in cancer, why do you think progress has been so slow?
Well, it is easy to get taken up in this, get excited and motivated by certainly the early studies. And I did some of the pricing and market access studies for the earliest CAR-T product, and US payers, the most stringent of people to convince, seemed quite positive on this when we presented the profile to them and the clinical data, because the first indication was pediatric ALL, and cancer, whatever leukemia or cancer you treat, it's not a nice thing, but in younger children, it tends to be easier to cure than in certainly older adults. So they were not so much bowled over, because payers don't get bowled over. They just, they nod and accommodatingly in some respects, but they were amongst the most interested in paying a reasonable amount of money for that sort of therapy. And that's one thing we haven't talked about. These drugs all in, or these treatments, should I say, are hundreds of thousands of dollars, $460,000, $450,000, $470,000 per course of treatment, which is no mean feat. And the reason we were explaining to payers was because certainly in pediatric ALL patients, you're actually saving the cost of a stem cell transplant, which in itself is curative. And you look at the early data from the CD19s, and on amongst the patients, that was curative. The patients were cured over the extent of the clinical studies. Now, later on, a number of issues have come up, and you talked about the positives of using a co-stimulatory domain in pairing the constructs, but the negatives are that, as I say, even if you're not a virus or a cell infected by a virus, you come up against an activated T cell, and it's not a nice experience. There are lots of toxicities associated with, for the patients themselves, like cytokine release syndrome, which in the early days of T cell therapy were actually fatal. The toxicities were worse in the short term than the disease. So there are some downsides. That doesn't make them a panacea for
But we have also learned to manage those
exactly
toxicities. Yep. Stefan Grupp, if I'm getting his name right, from UPenn, I think was the person who conceived of anti-interleukin 6 therapy, which was a major advance in terms of the safety. And I know with clinical experience, they've certainly learned to manage them much better than they used to manage those itis effects.
Absolutely. Anti-IL-6, and you take that, and in a pandemic as well. I mean, we'll do a podcast on anti-interleukins one day, because but they seem to be a panacea for a lot of things as well. But back to CAR Ts, yes, I mean, steroids, anti-interleukin antibodies have made that cytokine release syndrome much more manageable, and in fact they're prophylactically used, I think, now in many treatments. So yes, it was but we're getting back to, from the payer's perspective, and the commercial success or not so much of the CAR Ts is they cost a lot of money. And in pediatric ALL patients, we justify that by saying, well, it saves the cost of a stem cell transplant, which is about $500,000. And I read an article a few weeks ago that said effective CAR T therapy in pediatric ALL patients enables them to go on to a stem cell transplant, which is great, because patients get cured, but you're actually then not saving that $500,000 of stem cell transplant. You're enabling that. So it makes that treatment doubly expensive. So partly one of the things limiting CAR Ts is their cost. And that's because these are not easy things to do, right? You can't just put a few chemicals into a reactor and purify them and come out with a drug active substance. You have to take the cells out from the patient, transport them to a registered laboratory, do the transfection, amplify them up, purify them again, wash out all the vector, and transport them back and get them back into a patient. And as we're talking about with the steroids and the anti-inflammatories, but before they do that, you have to pre-treat patients to get rid of the bulk of the circulating tumors, circulating lymphomas. So it's not a simple thing to do a CAR T. Only a few centers worldwide even can do it.
Yeah. And I mean, that whole question of whether it was going to be a bridge to transplant or a replacement for it was sort of an open question. But I think with the 5-year survival data that Novartis put out recently, I think is about 55% of patients have surviving at 5 years. And these are
which comes back to a problem, right? So if you're a payer, how do you decide which patients to pay for? The ones that, the half that will respond and be curative, and the half that won't. And it's a difficult call, isn't it? Especially when you've got to make that payment, like the gene therapies we talked about before, up front.
Yeah, but it's just, I haven't looked at any more closely than that, so I have no idea. You obviously have to make the payment, and I don't know whether they can spot it early, whether or not which patients are going to relapse, and that they can potentially maybe move to transplant if that's required, if that's possible.
Well, I think you're hinting at the commercial success in recent times of these therapies. The big pharma companies that are now marketing those approved therapies, whether it's for pediatric ALL or B-cell lymphomas or multiple myelomas. And multiple myelomas is an interesting issue anyway, but whether the costs associated with it, the amount they can get back, and whether payers are being, seeing the real world efficacy and the costs associated with it, whether payers are now a bit more savvy in terms of, as they are for gene therapies, not paying up front. They're saying this is an outcomes-based reimbursement.
Yeah.
We'll pay you a portion every year for the patients. That may be what has led to the fact that in most big pharma companies that have a CAR T therapy, it's 1% or less than their quarterly revenues.
And sales of the long-established products seem to be slightly in decline as well.
Yeah. I mean,
high single-digit percentages, yeah.
Yeah, on an annual basis, and even from the longest ones on the market, most quarter to quarter. In fact, where they have been a success, strangely, is not in big pharma, and partly Gilead probably with their product is, yes, Carter is still growing quarter on quarter annually, but probably part of that success is that in a smaller biotech company outside of big pharma, then it's a smaller proportion of their revenues. Gilead has a track record in HIV and hepatitis C, two areas that are not growing that much anyway, so it might actually make their other products look quite good in terms of the magnitude and the growth.
Solid tumors have unfortunately not yielded anything, it seems to me, despite all the efforts.
Panacea rock hits a hard place of solid tumors, and they're difficult. I mean, you'd think that a hematological tumor with the cancer cells circulating in the system is probably the best scenario for an activated T cell population to go around infused and go around and take on. To get into the middle or even the periphery of a tumor cell, which is hypoxic, it has a whole range of antigens that aren't perhaps what the T cell is looking for, is a different kettle of fish.
Yeah, it has, immunosuppressive macrophages, it has immunosuppressive checkpoint antibodies being expressed, or no, checkpoint receptors being expressed. And that brings us back to, again, to the Rainier Brunton's quotation, because I mean, he made the point then, and this is 2017, for the second time here, folks, exclusively, that basically the heterogeneity of antigen expression in tumors means you can't just easily target them without having maybe catastrophic effects on healthy tissues, because you just don't necessarily have these uniquely expressed tumor antigens that make for clean targets. It's a huge problem. But the immunosuppressive milieu of the tumor microenvironment, which everybody was talking a few years ago, regardless of whether it was CAR Ts or antibodies or whatever the hell, about making cold tumors hot. It just this kind of mantra, because every single company in biotech apparently at one point was an immuno-oncology company. And this was as much due to the success of the checkpoint inhibitors more than the CAR Ts, really. But all that notion of making cold tumors hot, it seems to me that they're still cold.
But it is interesting. And you put out a very interconnected critique of cancer therapy today. These are not easily things to treat. We do have reasonably good and certainly new treatments for solid tumors. And as you said, the checkpoint inhibitors, the anti-PD-1 antibodies, which are almost exclusively used for solid tumors. At the same time, we have CAR Ts, which are only used for liquid tumors. I'm sure they didn't design it. It'd like to be a panacea for all, but we have it. Human ingenuity is good enough to actually invent and develop new treatments for those two parts of cancer in the last few years.
And on top of those two classes, we also have the antibody drug conjugates, some of which have almost CAR-like levels of efficacy without necessarily all the tox concerns, although I guess they do have their own tox concerns. But there's been some spectacular success in the ADC space, as we call it. And arguably, that's a kind of a hotter space right now than the CAR Ts in terms of cancer, at least. And then radiopharmaceuticals, there's been some spectacular progress there too. Again, it's an area that has its challenges in terms of obtaining secure supplies of isotopes. There has been some real progress and real investment there too. And again, both of those areas would have a big focus on solid tumor indications.
And both of those areas are going through something of a renaissance at the moment.
Yeah,
I mean,
yeah.
you and I both know that antibody drug conjugates go back to Mylotarg in the old days, and that was a horrible drug, eventually taken off the market because it was so toxic. Now we have the salad days of AstraZeneca's Enhertu, which has improved significantly on Roche's anti-HER2 antibodies. So we have, and interestingly, we've got an Achilles heel, if you like, of the CAR-Ts in multiple myeloma, because yes, the CAR-Ts were developed and show similar like responses to the earlier anti-CD19 CAR-Ts in multiple myeloma, but we've got good antibodies, good naked antibodies. We've got good bispecifics recently launched. And that's one of the issues that you talked to earlier, that's sort of holding down the commercial progress of the CAR-Ts for multiple myeloma. There's less toxic, more easily available or more easily administered drugs to treat multiple myeloma than there has ever been before.
But then there's two other allied fields in which the CAR concept is being applied. One is trying out CAR T cell therapies in autoimmune disease. And the other is attaching CARs to different cell chassis. So instead of using a CD8 T cell or a CD4 T cell, you're using maybe a macrophage or a natural killer cell in particular. And there's been early clinical data, and it remains early on several fronts there, that I think is very interesting. And in terms of the natural killer piece, a lot of the credit goes to Katie Rezvani of MD Anderson Cancer Center, who a couple of years ago, quite a few years ago, 2020 at this point, reported in the New England Journal of Medicine the use of CAR transduced natural killer cells in CD19 positive lymphoid tumors. And so this was non-Hodgkin's lymphoma, chronic lymphocytic leukemia. She used NK cells that had these CARs, these chimeric antigen receptors that recognize the CD19 receptor expressed by the B cells that were cancerous. And I think it was an early stage trial. She got 11 patients treated. 8 of them had a response, 73%, and 7 of them had a complete remission. That was pretty spectacular. And that elicited a huge pile into natural killer cells. They hadn't been industrialized as a platform as readily as the T cells had. They were a bit trickier to work with, and you had to put in a few different switches, IL-15, interleukin 15 especially, to switch them on and get them going. And they're just a little bit trickier to work with. It just took a bit longer to work out the protocols by which you could make them work. But that was very, very promising work.
Oh, it's taken a lot longer than that, Cormac. Ask any venture capitalist if they've ever invested in a company with NK cells, and they'll probably all, well, if they were honest, they would admit to, having been there, done that 10, 15, 20 years ago, and it didn't work out. I really hope it works out for CAR-Ts or NK CAR-Ts, whatever we're going to call them. But I also am fascinated by how we go around the block again and again in autoimmune disease. One of the longest and most successful antibodies, although it's now biosimilar, the CD20 antibody originally from Roche, Rituxan, started off as for non-Hodgkin's lymphoma, then found investigator-sponsored studies where it worked in multiple sclerosis because it takes down the cells that produce antibodies and produce an autoimmune response in multiple sclerosis. And there's a second-generation version from Roche, anti-CD20 antibodies. And now we have CAR-Ts, anti-CD19 T lymphocytes, going after CD20 antibody-producing B cells. It almost gets convoluted, doesn't it, between the CDs, the cells, the antibodies, the cells. Yeah, as long as the patients respond, and they seem to be, again, in early studies in multiple sclerosis and in lupus, it seems to be doing some sort of effective job.
Yeah, and I mean, we're all stuck with human biology. And so the themes keep on recurring because the diseases are still the same diseases, and we're still trying to find ways to effectively treat them. So where the anti-CD19 CAR T cell therapy seemed to work in lupus, whereas an anti-CD20 antibody didn't. Now, that said, it's early stages yet in lupus, and we still need long follow-up data. But it was posited, at least, that maybe it was better tissue penetration, and that I suppose you don't necessarily have an immunosuppressive situation when you have autoimmune disease. You certainly have an autoimmune attack on healthy tissues.
Yeah.
You don't necessarily give out the immunosuppressive signals that cancers do. So it's a different ballgame. But the work reported, it was from Erlangen in Germany, Georg Schett. This was a paper in Nature Medicine, which was published just this time last year, so October 2022. Again, in a small number of patients, they got clinical remission. They got, and the remission was maintained over a pretty long time.
Yeah, that's the important thing, I think. These T cells that are engineered, regardless of what the construct is, and even if it's more non-specific, to go after B cells that produce antibodies that give you an autoimmune response anyway, it's long lasting, right? They circulate in the body for ages, as long as they don't get exhausted. So you could effectively have quite an effective cure, which brings another sort of problem with my payer's hat. And how are you going to pay for it? And this is probably the crux of where we are in CAR T today. Effective therapies, but how are you going to pay for it?
Well, actually, I mean, if you look at, I actually think autoimmune disease is a fantastic test case or use case for CAR T therapies, because if these are curative and you give them a one-off shot, and yes, you do an outcomes-based pricing approach, and if you get people who are off drug therapy for life for $400,000 for the sake of argument, that's actually saving the payers a ton of money in the long run, if they're also otherwise getting biologic therapies. And I mean, there's been such a pile in the CD19 space for lupus, crediting Asher Mullard, Nature Review's drug discovery. He had a recent piece published last month. Like Novartis is in here, Bristol Myers Squibb, Cabaletta Bio, Kyverna Therapeutics, JW Therapeutics, BioRay Laboratories, Galapagos, Autolus, et cetera, et cetera, et cetera. There is a list as long as most people's arm of companies who are chasing the lupus opportunity now. And again, I mean, it's such a pile-in. It looks like that there will be casualties. It's rare to see this level of response so quickly to one tiny academic study. But lupus has been a really, really difficult indication, mainly affecting women, not entirely, but mainly women. And it hasn't had the same therapeutic successes as other chronic autoimmune diseases, such as MS or rheumatoid arthritis or psoriasis have had. And I think if this worked, it would be quite an extraordinary outcome, actually.
But I share your enthusiasm for it to work, because lupus is a forgotten disease, if you like. It's one of those things that's so difficult. Oh, let's not bother, or let's not waste R&D resource on it. But if you can crack it, then it could run straight into the paradox of global pricing and reimbursement, in that in European markets, where the arguments you say, you take a patient off expensive drug therapy for life, that's an economic saving because they go back to work. That's a no-brainer in European markets to do that. In the US, where people have commercial health insurance and they change health insurers 2 to 3 years, how are you going to get the payment for that if you're the original health insurer that pays that installment? How are you going to follow those patients around? That's sort of the paradox, because in Europe, it's an obvious no-brainer. In the US, you might, if you're a payer, you might lose the income stream. So are you going to pay for it? It's a almost developing the new therapies is not the hard part. Getting paid for them is, or making a profit out of them is.
Yeah, arguably, we shouldn't even be having this conversation, in the sense that if the thing works and patients need it, let's find a way to make sure they get it. And if the US reimbursement system ties itself up in knots and prevents patients getting access to therapies that they should have access to, well, that's just wrong and it needs to be fixed. There might be, I mean, it's not very scientific and it's certainly not very insightful, but it's kind of the plain person's response to that kind of very tricky scenario. And then we haven't got into all the various kind of synthetic biology, logic gated switches that the basic CAR Ts have now evolved into these kind of little, I won't call them robotic devices, but they have an awful lot of smart technology built into them in certain cases. They haven't all fully worked out by any means. But I think, to come back to our original sense of wonder, biology, T cells and other immune cells are programmable, and we can insert pieces of DNA into them to get them to make things that function in all sorts of extraordinary ways. And we can add these little safety switches to turn something on. With an easy drug therapy, you can add safety switches to turn them off. activation switches, rather turn them on, I should say. You can also do very interesting tricks where you can actually have a two-step antigen recognition method using leucine zip finger proteins. Senti Biosciences were doing this quite a number of years ago in really clever ways, so that you can sort of modulate the affinity and therefore hopefully as well dampen down some of the sort of toxicities that we saw, and also multiplex kind of your recognition mechanisms so that you might be able to target a cancer cell by saying recognize antigen X and Y, only if they're co-expressed. Or you can also say recognize antigen X and Y, but not Z. So if X, Y, and Z are present, you don't recognize that, but you only recognize when X and Y are uniquely present. So there's all sorts of extraordinarily interesting ideas, not all of which necessarily will make it to prime time. But I think that we are still at the early stages of innovation here. Now, it remains to be seen whether or not the funding spigot will remain open to maintain the innovation. But I think there's been so much activity here, and there's been such great success with the CD19s that maybe it'll be the CD19s all over again in autoimmune disease. But I think I wouldn't write them off, I suppose, is the point I'm making. Just yet.
All the plethora of technologies that you've talked about, in addition to the more recent ones, I'm sure you, like me, sometimes wonder, I wonder if I'll get cancer one day. And I don't have any worries now after this discussion. There'll be a cure there for it.
Yeah, I wouldn't be that quite optimistic. I still make sure that you get to keep up the old healthy diet and the good exercise and all the rest of it.
Oh, okay. That's boring. All right. Okay.
On your bike, Andy.
Yeah, as usual.
Until the next time, thank you.
Bye-bye, everyone.